Okay, I'm going to introduce to you Dr. Michelle Krishnan. She's going to be speaking to us from Roche. So Dr. Krishnan is what she described herself as passionate about translating cutting-edge scientific research into treatments for individuals with rare disease, and she heads up the rare disease group at Roche, and she has been involved in the Angelman community for the last 3 years, if I recall. And so we welcome Michelle back from Europe, and it's always a pleasure to see her. And she's going to talk to us today about Roche's endpoint study called the FREESIAS Study. Let's go forward.
Hi everybody, and thank you all for the invitation to be here. It is my 3rd year, and every year it gets bigger and every year it's more exciting, so I'm very glad to be here with you today. So my name is Michelle Krishnan, and I'm here on behalf of the Roche Genentech team to give you an update on our drug development program, where we are, and also to speak about the FREESIAS Endpoint Study. And you'll see some lovely balloons around the room that are relating to that as well. So that'll be the topic of what I'm speaking about. So the disclaimer regarding the opinions being my own. And so the first part of this talk, so there'll be 2 parts. In the first part, I'll be giving you an update of our drug development program at Roche Genentech. And just to make it clear, so in the US, Roche is also known as Genentech, so we're the same company. And so firstly, I will talk about our LNA molecule, and then after that, I will talk about how we move from scientific concepts to clinical trials. And then in the second part, I will talk about the FREESIAS study. Our approach to rare disease drug development at Roche is really to start with the science, and you've heard a lot of fantastic science this morning and continuing throughout the rest of the day, and really that's where it all starts. But we have to really work together with you and with the community to understand the disease in all its different ways and to understand Angelman in all its aspects, so that we can actually work towards the ultimate goal of bringing breakthrough therapies forward, and that's where we're all going to, and as I think was mentioned earlier, the competition is really the disease, and so we really want to work together with the community to be able to bring that forward. So the approach that we're taking is an RNA therapy. So you heard earlier today, and there was a beautiful video that Alison showed regarding the ASO approach, so it would come into that category. And so an RNA therapy is a molecule that binds to the RNA. So RNA is the stuff that eventually makes protein, and protein, as you know by now, is the building blocks of what happens in the cell. So RNA therapies can do a number of different things. They can reduce, modify, or increase the gene expression. And we actually have examples at Roche of using this approach in various other indications, other diseases that are further ahead in their development path. So in Huntington's, we partner with Ionis to work on an RNA therapy that is inhibiting protein production. In SMA, spinal muscular atrophy, we're actually partnered with PTC Therapeutics on a modification which is bringing real transformation to that pediatric disease as well. And for Angelman, we have an LNA. So an LNA is a modified version of an antisense oligonucleotide. So it's a modified ASO that modifies the expression of UB3A. I won't go through this in detail because it's actually been covered very well already today, but LNAs are an RNA therapy that can activate the father's UB3A gene to actually make UB3A in the brain. So as you've heard already, this will work by actually activating the paternal copy, so the father's copy of the gene, which is there in the neurons, but it's kept silent, and it's kept quiet by this antisense. And so an LNA therapy, which would be similar for an ASO, is designed to work in Angelman, since of course you know there is a genetic change on the maternal copy, on the mum's copy. That means that there is an absence of protein being made from that, and so we can actually activate the father's copy to produce UB3A protein. So the idea would be that that would bring back the expression of UB3A, of course being one of the core factors in Angelman. So this is an overview of the drug development steps that you've seen in a number of different diagrams over the day today. And it goes from discovery, so the idea, the scientific concept, towards the experiments in animals, in various species, in mouse, in monkey, and then moving towards the clinical stages of development. So towards trials from phase 1 onwards, and towards the availability of a new medicine, which is actually where the end goal is for all of us. And I'm very excited, and I'm very glad to be able to share with you today that we do plan to be able to start our phase 1 study in 2020. And so that's incredibly exciting for us. And so there are many things and complexities to the development of clinical trials, as you will become aware of. And so that is our hope, that is our plan. We will be coming to you with updates of how that moves forward and keep you updated on that. And the other thing to also highlight is that this is a long road. So this is the first step. These are the steps on that road, but this will be the beginning of a journey that we are on together. And this is just the very beginning stage for this community. So it's very exciting, and keep your energy and strength and courage for the road as we go forward together. How did we arrive to this point? So there's a lot of pre-work that happens in the discovery stage. And so we actually started with a human model of Angelman based on Angelman cell lines, so cells from Angelman patients that were actually then turned into neurons in culture, in dish. And the benefit of this is that it allows us to test many molecules. So we've actually screened over 2,000 LNAs to be able to arrive at the selection of the one that we're planning to take into the clinic. And that LNA has been designed in a very specific way and has been filtered according to its potency, so its strength. And that is measured in terms of its effect on UB3A. So you see at the top, essentially, what's going down is the antisense, and then in the middle, what's going up is the mRNA, so the thing that makes the protein, and then what you see at the bottom is that the UB3A protein also goes up. And that was in the dish with the Angelman neuron cultures. And then we looked at whether there were similarities between the results from human cells and monkey cells, because that's very important to help us understand whether we can learn things in monkey that are useful and informative of patients. So that's what you see here, is that actually it behaves similarly. So the results are similar when you look at monkey cells as well as when you look at human patient cells. And then we progressed forward to the next step, which is how do we look at this in an in vivo, so in an in-life setting in the monkey. And so here I want to pause and give a shout out and acknowledgement to a fan who actually was listening so carefully to the talk that I gave with my colleague Ina at ASF in the summer, that the person really remembered this information without me actually showing the plot. So I really want to call that out as a point of attention, that this person really remembered this. So thank you for listening so carefully. And there was a question around that I will try and answer now. That is one of the questions that was submitted to the Q&A panel for later, which was, what does it mean? So I mentioned at that time that after a single dose of the LNA, that we had seen a response after 85 days. And that's based on this plot, so I'll walk you through it. So what you see in the red line is the antisense, so the thing that stops the expression from the paternal copy. So that's going down. So after one injection of the drug at the baseline, this is an experiment in monkeys, okay? So that's very important to note. It's a specific situation. So we give a single injection just to understand better how the LNA is working and what its properties are. So after that one injection, the antisense goes down, which is the red line, and the blue, so that yellow line is the mRNA, so the thing that makes the protein, and then the blue line is the protein, and those go up. So the antisense goes down, the mRNA goes up, and the protein goes up. Now, I think a very important thing to highlight here, which came out in the question. So the question was, if we give a single injection of the LNA, will we see benefits in clinical improvement at 85 days? So we will not be able to answer that question from these data. This is an experiment in monkeys that are wild-type monkeys, and a single injection is given to understand the properties of the molecule. I think you all know that in the clinic, the ASOs and the LNA would require repeated administration, okay? So it would not be a single injection. There would be multiple injections required over a period of time, okay? So this is the example of these data. And from here, what we can say is we see these changes in the protein, which is really what we were looking to learn and demonstrate, which is very important from this experiment and is very encouraging because that's what we need to see. How that would translate to clinical benefit is something that we would actually all need to learn through the clinical trials that we plan to run in the future, and that would not be answered in this particular experiment. So I hope that's clear. I'm happy to speak to it further in the panel if there are still questions around that. So moving from this scientific component, what do we do with this insight and this understanding that we can generate this protein? How do we actually move to clinical trials from this? There are a number of steps on that path, and it's only really through this that we will be able to understand how to design studies well. And Emil Kakis gave a very beautiful talk about that this morning regarding the steps of clinical development and previous examples. We need to understand Angelman. We need to know how to measure changes in response to therapy so that we can design trials better. We need to understand what's meaningful to you and what we need to measure that's actually important. We need to know whether we have enough data on the safety and the general understanding of these different molecules, and we need to work together to do that. And the most important people are you in the room, and together with the scientific community and together with the clinicians and investigators and also our industry partners. And we are trying to work together with other companies to make things as quick and efficient and insightful for all of you. So we started with a disease concept model to understand what's important to Angelman families, and this was a lot of interviews with families, with caregivers to understand what would be worthwhile changing with the therapy. And so families said that expressive communication, independence, sleep, maladaptive behavior, gross motor, fine motor, and seizures would be important things to change. So this disease concept model is very extensive, and the full report is available to you through FAST or ASF, but we are planning to publish this as a scientific manuscript as well. So when we then looked at these concepts that are important to families and mapped them to scales that are being currently used in the clinic and in other clinical trials, you can see that there are some gaps in terms of what's important to families, but that was not being measured very well with the existing scales. And sleep and seizures were particular gaps. And so those being very important things to measure and understand, we need to actually get better knowledge on those particular aspects. And that is where we've actually been working to bring together the FREESIAS study, which is a very important piece of this development path. So I can tell you more about that now. It's important to note the FREESIAS study, which is a study that is currently running, is a non-drug study. I want to make that really crystal clear. So that is an observational study. That means there is no drug involved, but it is a very important study because it helps to understand how we can actually design clinical trials for Angelman in the best possible way to actually answer the questions that need to be answered for the community. So it's a study that's being sponsored by Roche Genentech, and it's co-funded with our collaborations with Biogen and Ionis. So the companies are working together so that Roche isn't running a study, Biogen's running a separate one, Ionis is running a separate one. We're actually doing that together so that it's more efficient. And we are also sharing overlapping data with the FDA-funded natural history study and also with an MRI study that's happening at UNC. So this is all to be able to facilitate families taking part in all these studies without it being a conflict and without it being an additional burden, and so that the data can be of the greatest value to the community. So the FREESIAS study is an endpoint study, and a lot of it happens in the home, and there are a lot of digital approaches, digital devices that are new, and it's a collaborative approach. So it runs over currently 1 year, and there are 2 clinic visits, one at the beginning and one a year later. And there are 3 home visits, and those are really important because it's actually where there is a home assessment of EEG overnight, which is done by a specialized company called Neurotech that have been doing that for many years. And the purpose of it is really to look at the feasibility. So is it possible to do a lot of these new things that haven't been done before in clinical trials to be done in Angelman? And there's a lot of questions around what's possible. There's a lot of exciting new ideas, but is it actually really feasible for families? And so that's an important part of understanding. We are looking at recruiting Angelman patients aged 1 to 12, and there's about 40. Also about 10 Angelman adults and about 10 to 20 controls. And it's really about looking at this feasibility and hearing from you what's working, what's not working. So how will this help to develop future trials for Angelman? So it's really about endpoints. So endpoints are the things that we measure in a trial that will tell us whether a drug is actually being beneficial and making a difference to how someone feels and functions or not. And we're trying to do this in a more quantitative way, so things like the digital devices that are more accurate ways, potentially, and more realistic ways of measuring what's happening rather than relying on a single visit to clinic. And it's a small, so how is this different to a natural history study that might run for many years and which is also a different type of study? So this is a small number of individuals. It's over a short set period of time, and there's a lot of very in-depth assessments using these new devices and looking at EEG and sleep in the home with the intention of really designing future trials with the minimum burden and impact to families in terms of the scales and assessments that you're asked to do in the future. So we're focusing here on these building blocks that are highlighted in orange. So there's a number of different things you can try and learn from natural history studies. This is a different type of study. So it's an endpoint development study, and it's about informing the design of clinical trials. Potentially, the data could in the future be used to supplement placebo arms or even substitute placebo data, depending on how the discussions go with regulators. There are these options to use these data as baseline or as information for future comparison for studies, and understanding what's happening in the absence of drug to help get a better understanding of the safety profile of that drug in the future. Happy to say that we have a number of sites open across the US, and you see them here marked on the map. So we have sites at those various states, and we are planning to have 3 additional sites across the US that will be opening very soon. And we've only been recruiting since about September this year, and it's amazing to see that we've already had 13 families join, and of those 13 families, 12 are angels, and so that's a really fantastic testament, I think, to the interest and support and understanding of the value in the community and among families of the value of observational studies and what this could bring to the community. So really a tremendous thank you to all who are involved, and really appreciate your dedication and commitment to be engaged. So we wanted to bring something back to you as soon as we could. Really, this is very, very fresh off the press. I had to really prod our biomarker leader and say to him, we have to take something. So we've actually put here some real data that have come out of the study, just very, very recently. And we haven't even had time to analyze these, but we just wanted to bring this back to you. And what you see there, so those wiggly lines are EEG traces, which many of you may have already seen from reports or when you've been in clinic. And so these are from patients in the study. So those photos are stock photos of what it looks like to have it applied, and many of you have seen that being done in clinic. So this is done in the home. So it's a full EEG setup that is set up at home to record EEG overnight, because we know that sleep is so important and so disrupted in Angelman. And it's fantastic to see that we're starting to see that it is feasible to collect this type of data in the home with Angelman families, and that's really important to learn. So we've got good data sets, it seems, from a number of the families that have joined. Not all of them have had the opportunity for home visits yet, because it's still very early in the planning of the study. So that's really fantastic feasibility information to be getting already, which is one of the really big learnings that we hope to get from the study. We're also getting nice data from the sleep mat. So this is a sleep mat that we've chosen because the raw data can be accessed, and it's a slightly larger device, so it should cover the bed area better than some of the smaller devices, so that when people are wriggling around, that it could still capture some data. And we also have a number of dedicated diaries that we've developed specially to collect data on seizures and sleep during the study. So these are all really exciting signs that it is going to be feasible to look at some of these new questions that could be very informative for whether a medicine in the future could be helpful to the Angelman families. So for the FREESIAS study, which again to emphasize is a non-drug study, but it's an observational study that can be extremely informative. So if you would be interested in joining that, you can get more information from our trial support line, and there's a number that you can call there. There's also an email also that can provide information. And we have brochures with all this information at our booth, which is at the back. And there's a number of our lovely team members who are there most of the time. And I will be there tomorrow afternoon as well, after 3 o'clock. If people want to come and chat, I'll be around. And also a really big shout out and thank you to FAST, to ASF, who've been with us from the beginning and really appreciate all the input and all the guidance and discussions on what makes sense, what doesn't make sense, how to try and design these types of studies in a way that actually is doing what we believe it can do to help design future trials. So in summary, I've touched on a number of building blocks for clinical development. So starting with the early development and discovery and how you develop a molecule, but then in terms of the clinical development, the disease concept model, the observational studies, also a shout out to our friends from the Global Registry, which is a really important really important source of knowledge. So you all are in the room, and it's fantastic to have so many people here, but you all know, and we all know, there are many people who are not able to be here today, and they are just as important as everybody who is here, and the registry is a really fantastic way to get that information about everybody, so that we can design trials that are fit for everybody. There's also the clinic database, which is a new resource that's being started up now and will be a tremendous resource as well. And this should all be done collaboratively, as I think we can't emphasize enough. So again, thank you to all the families. Also, thank you to all our site teams already for the FREESIAS study. There's so much work being done by study coordinators, sites, evaluators. So thank you to everybody, and to all our academic collaborators and investigators at the various sites for the FREESIAS study. That is a tremendous contribution from everybody already. So thank you very much, and happy to take questions. Off the hook.
So if we don't have any questions, you can save them all for that. You answered the big question that everyone had, which is wonderful. So thank you for
I
saving
read
us
your
time.
instructions.
I did. Thank you. You're the only one.
Okay, thanks
Thank you
everyone.
very much.
Happy to see you.
We have one question.
Oh, we have one?
Oh.
Yes, you dropped that bomb about the clinical trial thing. So what would the age criteria be for that?
For which one, sorry?
For the, not the FREESIAS study.
Oh, okay, the phase one. So
what are you
Alison's-
thinking?
Yeah, again, Alison is always on the ball. So for today, I wouldn't be able to answer your question straight, so I'm not going to try and talk around it. So what I can share with you today is about us aiming to start in 2020, and then what I can commit to is that we will keep you updated in real time about what's happening. So as soon as we can share that information with you, we will do that.
Okay.
Yeah.
Thank you.
Anything else?